or base has a very different kind of effect. So generally speaking, optimal pH for most of our enzymes is right around 7. It's a neutral pH. But as we diverge to a more acidic or a more alkaline environment, that enzyme activity is going to denature because acids and bases have the same sort of effect on our proteins, on our enzymes. And then there are some that have an optimal pH that's much closer to those that live in our stomachs. Okay, so enzymes are great because they help us to catalyze reactions. And catalyzing reactions, we don't always want to catalyze reactions, right? There's sometimes when we don't want that reaction to be active. And there are a couple of ways for us to do that. There is a really cool type of regulation called allosteric regulation. And with allosteric regulation, we get an enzyme whose active site is changed by some other molecule binding away from that active site. Now, this can affect an enzyme in one of two ways. It can either repress enzyme activity or it can activate enzyme activity. So with allosteric control, what happens is we have an enzyme that has some active site here at the middle. And then away from the active site, there is another binding site. Allosteric control means that there is a control molecule that binds in here at the allosteric site. And that allosteric site affects the active site in some significant way. So with allosteric suppression, what happens is the default position for the enzyme is active. That is, when there's nothing bound here to the allosteric site, that enzyme can catalyze the reactions that it's intended to. But when an allosteric molecule binds to that allosteric site, it changes the shape of the active site so that it can no longer bind to the substrate. It's a way for us to deactivate an enzyme. Alternatively, we can have allosteric activation, which means that the enzyme's default position is inactive, and we only activate that enzyme with an allosteric molecule that binds to the allosteric site. When that happens, the active site reclaims its active position, and it can start catalyzing reactions. Alternatively, if there is an allosteric control, there can be something called competitive control. And competitive control means that there is a molecule, a suppressing molecule, that binds to the active site so that the substrate can't get in there. The substrate is actively blocked by a competing molecule. So those are the two broad classes of enzymatic control. There is competitive, and competitive control again means that a molecule is binding to the active site. A competing molecule binds to the active site. With allosteric control, we have a molecule that's affecting the enzyme shape by binding away from the active site. So there's a really cool feedback process with many biochemical pathways. Specifically, there's something called feedback inhibition. So what happens is sometimes the product of a chemical reaction becomes an inhibitor to that chemical reaction. And that inhibition can either be competitive or it can be allosteric. So I mentioned before that one of the products of cellular respiration is a molecule called pyruvate. Pyruvate is a 3-carbon molecule. When we're actively going through cellular respiration and trying to produce a whole bunch of ATP to support cellular activity, that pyruvate is immediately brought in to the mitochondria. And it's used inside the mitochondria to create a whole bunch of ATP. However, if our cells are inundated with ATP, that pyruvate isn't brought into the mitochondria. And that pyruvate becomes an inhibitor to the enzymes that are making the pyruvate. Okay, so let's think about this hierarchically, right? We don't always want chemical reactions to be occurring. And there's a couple of ways that we can inhibit chemical reactions that are enzymatically controlled from occurring. And that control can either be competitive or it can be allosteric. With competitive control, we're taking an enzyme and we're putting a molecule in the active site so the actual substrate can't get in there for us to break it down or to create something from it, right? With allosteric control, what happens is we take that enzyme and we bind something away from the active site so that that enzyme loses its shape and it can no longer interact with the substrate or the reactant. One way in which that inhibition occurs is by feedback inhibition. When we start making too much of some product, that product can actually inhibit further reactions from occurring. So this is a diagram of both allosteric activation and inhibition. So on this first one, we're inhibiting or we're repressing some enzymatic activity. So you'll notice that this enzyme has 2 sites on it. It has an active site. It's able to take a substrate and make some products from it. Exergonic or endergonic reaction. Exergonic reaction, because we take something complex and we've made something a little bit less complex from it. We're breaking something down. But what we can do, if we don't want that exergonic reaction to occur any longer, we can take a repressor molecule and we can bind it to that allosteric site that's away from the active site, right? And when that happens, it changes the shape of the active site so that that substrate can no longer fit in there, and that enzymatic reaction ceases. Alternatively, we can actually activate chemical reactions. So that's what we have diagrammed here, where again there's an active site and an allosteric site. But that active site is in the default position isn't a shape that accommodates our substrate. So the substrate can't get in there and that active site can't catalyze the reaction. However, when we bind an allosteric activator to it, that active site does achieve the appropriate shape and our exergonic reaction can move forward. Yeah, go ahead.
So are both the repressor and the activator both allosteric?
They are. Remember, the definition of allosteric is a molecule binding away from the active site, and that's occurring in both of these examples. The active site is down below, and we have some control mechanism that's binding away from that active site. Okay, so we're almost done. We'll get out a little bit early today. I can see that you're beginning to wilt a little bit. So bear with me for just a few more minutes. In our bodies, cellular respiration, the primary product from that process is a molecule called ATP. And I've mentioned it a few times, and we need to talk about what that molecule actually is just very briefly. So ATP is an energy molecule. Please just think of it as a package of energy. and it's a transportable package of energy. Now what I'm going to do is I'm going to draw a monomer, and I'm going to want you to tell me what that monomer resembles. So ATP consists of three things. It consists of essential sugar called ribose, and I hope already this is starting to sound familiar to you guys. And then attached to that central ribose, there is a nitrogenous base that is a purine. And a purine consists of a 5-member ring that has both carbons and nitrogens in it attached to a 6-member ring that also has those carbons and nitrogens attached to it, or that make that up. And that nitrogenous base is called adenosine. And then attached to that sugar on the other side, there is a large molecule called phosphate. It carries a negative charge. So what does that look like to you guys? Do you remember? Think back to biologically important molecules when we were sticking stuff together with dehydration synthesis. And the bond between these two adjacent monomers was a phosphodiester linkage of that clues you ended off. Because remember, so there are other variants of this monomer, right? Some of them can have thymine, some of them can have guanine, some of them can have cytosine, or we can, in another context, we can stick A's to T's and G's to C's. What was that monomer called? A nucleotide. Yeah, you guys, ATP is essentially just a nucleotide, and it's the A nucleotide that's floating around. But we take this A nucleotide, and we're going to switch it up a little bit. And the way that we switch it up is we take more of those phosphates, and we stick more phosphates onto it. I'm just going to draw it off to the side because I didn't leave myself enough room at the top. Let me try that. So, ATP is adenosine triphosphate. What that means is we have this nucleotide, an adenosine nucleotide, that we've stuck triphosphates onto, or 3 phosphates onto. Now we know that phosphates carry a negative charge. And in nature, do opposites or similars attract? In the terms of electronegativity. Opposites attract, right? So negative is going to be attracted to positive and positive to negative. And if you take two negative sides of a magnet and you try and stick them together, what happens? They push out against each other, right? So what we've done is we've taken 3 negative charges and stuck them all together with covalent bonds, which makes this incredibly unstable. And that last bond particularly, super high energy. The way that we store energy in ATP, basically, is with that last bond. We have to invest a bunch of energy just to stick those 2 phosphates together. And once that bond is broken, a ton of energy is released. So this is perhaps a better diagram of our ATP molecule. We have our ribose down here, and we have adenosine up here, and then we have 1, 2, 3 phosphates attached to it. And that's all ATP is. This is what we want to make from cellular respiration. This is what plants make through the first part of photosynthesis, ATP. So, making ATP is inherently an endergonic reaction, right? Because we're investing a bunch of energy to stick these 3 phosphates together. But when we use that ATP, that is inherently an exergonic reaction because a ton of energy is released. All right, questions about the structure of ATP? Yeah, Carly.
You said that, to hold the three negative suites together, it takes a whole bunch of energy, and most of that energy is stored in the last.
Yeah. This represents the most unstable bond, so holding it together requires more energy. I think of it like trying to put 3 of my daughters in the same room together. It just doesn't work. They all repel each other. Okay, we okay with this? All right. So, what we're going to do starting on Monday is talk about photosynthesis. And very briefly, this is what photosynthesis looks like. So photosynthesis is broken down into 2 reactions. And I'm gonna write the reactions up on the board again. So with photosynthesis, the overall reaction looks something like this, where we take 6 water molecules and 6 carbon dioxide molecules, and we produce 1 C6H12O6 and 6 oxygen molecules. So what is this? Sugar, exactly right now. Monomer, is it a monosaccharide, disaccharide, or polysaccharide?